The process of adult neurogenesis can be actually boosted by a number of different factors, but the strongest physiological way that we can boost our hippocampal neurogenesis is exercise.
Welcome, everyone, to Longevity Roadmap. I am Buck Joffrey, and today we're going to talk about a very fascinating topic. That topic is adult neurogenesis. What is adult neurogenesis? It's basically the brain's ability to produce new neurons throughout life, which, by the way, in medical school, I was taught that that does not happen. But obviously, we now know that it does, and so we have an opportunity to speak with Dr. Walker on how it all works, and what is this process, where it happens, how it declines, and all of that. Fantastic interview, a really good look into potential implications for research in cognitive health and neurodegenerative diseases, and we'll have that interview right after these messages. Hey, longevity enthusiasts, it's time to take it to the next level. I've been fine-tuning my longevity regimen for years, and I look better and feel better than I did a decade ago. In fact, my blood work is even better than it was back then. And it's all because of my data-driven regimen, and it's inspired me to create a course and community just for you. It's called the Longevity Roadmap, and I urge you to check it out. If you're tired of your belly fat, tired of being tired, or just want to optimize yourself for the next 50 years, visit longevityroadmap.com. That's longevityroadmap.com. Welcome back to Longevity Roadmap. Today, my guest is Dr. Tara Walker. She's a senior research fellow at Queensland Brain Institute at the University of Queensland. She's a neuroscientist with a PhD in neurobiology and a leading expert in adult neurogenesis, which is the process by which the brain continues to produce new neurons throughout life. She is internationally recognized for her contributions to our understanding of brain plasticity and how we might one day harness these pathways to prevent or reverse cognitive decline. Welcome, Dr. Walker. How are you?
Thank you. I'm great. Thanks.
Great. Well, to begin with, I think it's probably a good idea for us to start up with this concept of adult neurogenesis. For one thing, when I was in medical school, that was, over 20 years ago, we didn't really think of adult, having neurogenesis, right? We used to think of it as something that just never happened after a certain age. So maybe you could talk a little bit about adult neurogenesis and kind of maybe how it plays a role in maintaining cognitive function.
Yeah, sure. Yeah, you're completely right. Like when we were at school, we were sort of taught that once the brain reaches adulthood, it's static. No new neurons can be formed. They can just be lost. And so this was like the long-held dogma. It wasn't until about the 1990s when they first discovered that there is actually a stem cell population in distinct areas of the brain. So there's only a couple of the areas of the adult brain where neurogenesis actually continues throughout life. In the human brain, the main region is the hippocampus. So the hippocampus, obviously the region of the brain that's involved in a lot of learning and memory processes. So there's a population of stem cells that was discovered in the hippocampus in the 1990s. And it was from there that they then, that the process of neurogenesis was then really thought to happen. It was first mentioned in a few papers in the 1960s that there was potentially this new neuron generation in the brain, but this was basically largely ignored because they didn't understand how this could possibly be happening without stem cells being present. But now it's widely accepted that there's ongoing neurogenesis in the hippocampus of pretty much all mammals, including humans. And yeah.
So is that neurogenesis? I imagine it's probably like most things in humans tends to not do as well as we get older. Is that
Exactly
part of the issue? Maybe you could talk a little bit about how, age affects that neurogenesis.
Yeah, for sure. So yes, you're completely right. So the process of adult neurogenesis. So we have this population of stem cells in the hippocampus that's continuously dividing, produces these sort of proliferating precursor cells, which then go on to mature into newborn adult neurons, which are then functionally indistinct from the other neurons in our hippocampus. But this process does decline significantly as we age. And this is what can lead to a number of learning and memory deficits as we age. Neurogenesis is very highly associated with specific types of learning and memory, particularly spatial learning and memory. So our ability to navigate around in our environments using cues. So, for example, if you're walking around the city and you're looking at certain landmarks, your ability to navigate in that space is very much dependent on the process of hippocampal neurogenesis.
Well, and why is that? Because I would think if you've got stem cells coming from the hippocampus, why are they localized in their effect? Are they pretty much? I mean, you would think, or I guess I would think that they were sort of involved with neurogenesis throughout the brain, but it sounds like maybe some parts are more involved than others.
Yeah, so as I said, there's only the stem cells only present in the dentate gyrus of the hippocampus, and
Right.
so they only repopulate the hippocampal region of the
got
brain.
it.
They
So
don't
they don't
migrate.
spread.
No,
They're not like bone marrow going to blood and all.
No,
okay,
no.
Got it. Okay, well that makes a lot of sense. Okay, so then one of the things that you've talked about, I guess, or you've shown, is that physical exercise boosts neurogenesis. So what are those? What's happening there? What's the mechanism?
Yeah, good question. So
Yeah.
that's what my lab's been researching probably for the last 15 years or so. The process of adult neurogenesis can be actually boosted by a number of different factors, but the strongest physiological way that we can boost our hippocampal neurogenesis is exercise. So we have known for about 20 years or so now. It was first discovered in mice about 20 years ago that when we exercise the mice, we get a significant increase in hippocampal neurogenesis. But we didn't really understand the mechanisms by which this occurs. We still don't fully understand the mechanism, but my lab's been working on this for quite a while now. And we hypothesize that it must be sort of systemic factors that might be released into the blood after exercise, because exercise is sort of, full body systemic sort of action. We hypothesize that there must be something released into the blood following exercise that might be mediating this. And so for the last probably 5 or 6 years, we've been investigating possible exokines, so these factors released into the blood after exercise, as being mediators of this increase in neurogenesis. So some of these factors released into the blood can cross the blood-brain barrier and directly affect the stem cell niche, or some of them can actually work indirectly by changing other factors in the blood, which can then communicate with the stem cells. And we've discovered a couple of these factors, which we've been working on in my lab, that we can actually now show can act as exercise mimetics. So if we take a single factor that we've discovered is upregulated in the blood after exercise, and then deliver this factor directly in mice so far only, that we can actually replicate a lot of the sort of regenerative effects of exercise.
So as you mentioned, in part what happens with hippocampal degeneration, you mentioned spatial navigation, but also we often think about memory formation, right? Emotional regulation, things like that. Is it the case that when you exercise that you get this kind of neurogenesis that potentially you are improving or maintaining memory formation more so, spatial navigation more so? Is there any data actually showing that?
Yeah, so there is some. There's obviously neurogenesis isn't just contributing to spatial navigation. That's just one of the aspects. It's neurogenesis is contributing to a lot of types of learning and memory that are involved, that the hippocampus is involved in mediating. That's for sure. Yeah.
Right. And so I guess one of the fascinating aspects of your work is the discovery of these selenium transport proteins, right? Selenoprotein P. I don't know if that's how you pronounce it, but it plays a key role. Can you talk a little bit about how that works?
Yeah, so we discovered that was one of the factors I was just talking about that we discovered was significantly upregulated in the blood following exercise. So we did an experiment where we basically took some mice and either had running mice or non-running mice and looked at what proteins were changed in their blood. And selenoprotein P was actually the most highest upregulated protein in the blood following exercise. So we thought, oh, that's really interesting. Maybe selenium levels might somehow be mediating this response to exercise. So we've done a number of experiments. I won't go through all of them, but basically we could show that selenoprotein P selenium is required for the exercise-mediated response. So we had when we knocked out either selenoprotein P or its receptor, we had knockout mice for either of those. We could show that the response to exercise was no longer present. So basically this transport of selenium into the brain was required to mediate the response to exercise. And we could also mimic the exercise response when we actually delivered selenium to the mice alone. So we could give dietary selenium supplementation, for example, to aged mice, and we could reverse the age-related cognitive decline that we observed in those mice. And we could also increase neurogenesis. Yeah.
So age-related cognitive decline in general, or is stuff that you would think of specifically as related to the hippocampus?
Specifically related to campus is what we looked at. So we looked at a number of spatial learning and memory tests. Yeah.
Okay. Is there any reason? Okay, because listeners of mine are going to be very eager to go out and buy selenium, but but the question is, if you're already exercising a lot, is there any value in additional selenium supplementation? From what you could tell?
No, so I think as long as most people should be getting fairly good levels of selenium in their diet. So selenium is a trace element that's found in lots of commonly eaten food. It's basically in all primary produce because it's found in the groundwater. So it's in the soil and it's taken up into plants and animals via the soil. So as long as you're eating a healthy, balanced diet full of fruit and vegetables and nuts and grains and all these things that contain selenium, you should be fine. But selenium levels do decrease with age. We don't know why. So perhaps as we get older, we should be potentially taking some more selenium. Selenium is actually found at higher levels in Brazil nuts. That's the highest level of the highest dietary form of selenium. So basically, you only need to eat one Brazil nut per day to get your daily recommended dose of selenium. So
I think that's also what they say for testosterone as well, oddly enough. Brazil nuts, there's something odd about Brazil.
Yeah, well, they have very high selenium levels, so you don't want to overdose because selenium does have quite a narrow toxicity range.
Oh, okay.
So you don't want to overdose on selenium. So don't go out eating like a whole packet of Brazil nuts per day.
There you go. So do you, I don't know, maybe this is already happening, but do you envision selenium supplementation for, practical intervention for people with, for one, limited mobility, and elderly or people who've already have some kind of neurological injury?
Yeah, so that's a good question. We've actually, in the same study that we published with the aging study, we were also looking at stroke, selenium supplementation following stroke. We have published that paper and another paper looking both in mouse models, showing that the selenium supplementation can protect against ischemic stroke. So we're now actually starting a clinical trial here in Brisbane. We haven't started recruiting patients yet, but we're just in the very early stages to investigate selenium supplementation for ischemic stroke. So
now, again, I guess I'm going to keep asking the same question, but we're isolated. We're again isolated to essentially just to where those neurogenesis is occurring, right? The hippocampus. Yep. So even for stroke.
Yeah, so with the selenium, it's probably definitely having a lot of effects outside of just affecting neurogenesis. So we could show that selenium affects neurogenesis, but selenium is also a very strong antioxidant. So it's going to have a lot of other neuroprotective effects following stroke just due to its strong antioxidant capacity. So it's going to be protecting against a lot of the oxidative damage that's occurring directly after stroke.
Yeah, talk a little, if you could, talk about ferroptosis.
Yeah, ferroptosis. Yeah.
Ferroptosis.
Yeah, so we're actually very interested in that now. That's what my lab's been investigating for the last probably couple of years as the potential mechanism by which selenium might be conferring its effect. Because ferroptosis is a relatively recently described form of cell death. So when people think of cell death, they think of apoptosis, obviously. But there's now so many other forms of cell death that are being described. So ferroptosis is interesting in the context of selenium because the core regulatory protein that mediates ferroptosis is glutathione peroxidase 4, which is a selenoprotein. So it's basically the levels of ferroptosis can be mediated by the levels of selenium that are present. So we're investigating whether the effects that we're seeing of selenium on neurogenesis, for example, might be occurring by mediating the ferroptosis of the proliferating precursor cells that are involved in this.
So basically,
process.
slowing down the death of those precursor cells.
Yes, protecting a subpopulation of them from dying, so therefore getting an increase in net neurogenesis.
So you also mentioned that oxidative stress can lead to death of those precursor cells as well. So part of the idea might be, this is how you can mitigate that risk, right?
Exactly. So the process of neurogenesis is actually really interesting because you have these stem cells that are proliferating and they produce this really large pool of these proliferating precursor cells, but only about 50% of them survive. The other 50% die, just eliminated. And this was sort of thought to be apoptosis, but we are postulating that a lot of these are dying via ferroptosis and that we can rescue these via selenium or direct ferroptosis inhibitor drugs.
It's interesting, just sort of neuroprotective.
Exactly.
So you've also shown that signals from activated platelet factor 4 can also stimulate neurogenesis. How does that work?
Yeah, so from that same screen of the blood that we did from the exercising mice, the other really highly upregulated factor was platelet factor 4. And so we thought that's very interesting. That's the protein that's released by platelets when they get activated. So as you know, platelets are these small blood cells that are primarily involved in hemostasis and blood clotting. But they're now having like this sort of emergence as an interesting population of cells that might be mediating a whole bunch of other things. So we were very interested whether platelets might be also mediating things like the effect of exercise on the brain. So we did a bunch of experiments where we could basically deplete platelets in mice. And when we had platelet depleted mice, we could basically no longer see the effect of exercise in these mice. So platelets are somehow also mediating this effect of exercise. And the interesting part to sort of tie it all together is platelets contain like more than 1,000 like bioactive molecules. And one of the things that platelets actually can contain is selenoprotein P. So we're thinking that it's somehow all connected and that when platelets get activated, they release this like basically a plethora of proteins that could then be released into the blood and maybe mediating the effect of exercise.
And when you're talking about platelets being activated, are you actually talking about like a coagulation pathway? Like, what do you mean by that?
So they still get activated. So activators is when they basically release all the contents from their granules. But we did some proteomics on platelets that are activated after exercise and compared that to the sort of proteomic profiles of platelets that are activated in response to sort of wound healing. And their profile is very, very different. So the platelet activation that occurs after exercise leads to the release of a very different sort of subset of proteins that is released after activation in response to wound healing, for example. So we believe that we've identified sort of a novel arm of platelet activation and how this could be mediating things like response to exercise.
What do you know about maybe the relationship between chronic inflammation and the decline in neurogenesis? Are there inflammatory markers suppressing neural stem cell activity directly?
Yeah, so I mean, I don't know so much about this, but there's definitely all related. We're very interested also in looking at the role of microglia in mediating the process of neurogenesis, and they certainly are. Obviously, inflammation in the brain increases with age, and it can be decreased with things like exercise. So, and also we've shown. My colleagues and I have also shown that things like platelet factor 4 can decrease inflammation um, in the brain, which also leads to improved cognitive function in aged mice.
Yeah. So there's also, an increasing focus on cellular senescence in the aging process. Is there any evidence that senescent cells in the brain or the signals they release impair neurogenesis?
Yeah, there's not so much research being done on that so far. That's something that we're very interested in starting to have a look at as well. But yeah, that I agree. This is something that's sort of emerging more recently as an interesting topic, and I think they probably are. There's a couple of schools of thought as to why neurogenesis might decrease with age, and one school of thought thinks that it's because the stem cells are becoming senescent, and the other school of thought is that they're sort of depleted and eliminated. So we don't really
Yeah.
know the answer yet, and that's what a lot of groups are working on at the moment, trying to understand.
Yeah. So looking at your work again in the context of neurodegenerative disease, um, do you think or consider neurogenesis like sort of an emerging therapeutic strategy of stimulating neurogenesis, or more of a preventative approach?
I think you could look at it from either aspect. So we're very much interested in looking at Alzheimer's now in my lab. We're looking in mouse models of Alzheimer's disease, and certainly neurogenesis does decrease in neurodegenerative conditions like Alzheimer's. In humans, also, there's a few more recent publications that are showing that neurogenesis levels decline significantly with Alzheimer's disease patients. So yeah, we can look at sort of increasing neurogenesis in this context as a therapeutic, or alternatively, obviously, keeping your neurogenesis levels higher throughout life via exercise or enriching like constant learning and memory, all these type of things, is also beneficial. And we know obviously that things like exercise or lack of exercise is one of the major contributors to developing conditions like Alzheimer's disease. So,
Yeah.
and
Uh,
obviously neurogenesis only plays a small part in mediating that, because exercise obviously has lots of other roles like anti-inflammation and all lots of other things, as well as promoting neurogenesis. But that's just
right.
one part of
Right.
it.
Right. Interesting. Well, very interesting stuff. I appreciate your time. And where can people learn more about the work you're doing?
So basically, the best place to find my work would be on our Queensland Brain Institute website. So it's got a link to my profile with the link to all of our research papers, etc. there.
Thank you so much for your time.
No, thank you. It's been lovely chatting to you.
Thanks for listening. A quick reminder that while I am, in fact, a surgeon, nothing I say should be construed as medical advice. Now, make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe.